So what we did was we built a multi-center trial that was done in 3 centers in Canada, in London, Ontario, at London Health Sciences Centre, at the CHUM in Montreal, and also at the Jewish General Hospital in Montreal. And what we did was we used healthy donor stool to prepare capsules for a process called fecal microbiota transplant in melanoma patients. What we did was we brought in the patients after they underwent bowel prep, which means they effectively got rid of their own microbiome the day before. And this is similar to when patients go undergo colonoscopy. And then what happens is that they come in, and then we'll give them the capsules. And then 7 days later, we allow 7 days for engraftment, that they have the donor's microbiome. After 7 days, they started to receive a single agent anti-PD-1 therapy, and that could be either Nivolumab or Pembrolizumab based on the investigator's choice. And what we did was we first looked at the safety of this combination therapy and see how safe this approach was for our patients, because this was the first time that anybody has tried this with healthy donor material at the first line. So these patients were treatment naive, or I should say anti-PD-1 naive, for their cancer. And we saw that this combination treatment was quite safe. There was no unexpected toxicity from the FMT or any added toxicity to what we expect from single agent anti-PD-1 immunotherapy. And what else we saw in terms of translational analysis of the patient samples, first of all, something that was very interesting was that we saw the healthy donor microbiome had a much higher diversity in the donor microbiome compared to the patients. And we saw that the patients post-transplant, their microbiome came quite diverse as well, similar to donors really, and that was stable over time. However, we did not see this newly built diversity to be linked to response in patients. What we saw was that being able to retain the donor microbiome and having a successful engraftment and successful retention of the donor microbiome was key to the patient's response to immunotherapy, which further validates the significance of microbiome in really shaping and controlling anti-tumor immunity that is being leveraged by immunotherapy. And the other things that we saw in this study, and that was very important, was the presence of certain metabolites in the patient's plasma post-FMT that was really correlated with response in the patients who responded to therapy, and some changes with the immune cells that we saw the patients who didn't respond actually had certain population of their immune cells. These are mucosal associated T lymphocytes or mate cells, that they were more exhausted post-transplant. And the other thing that we saw was in our translational studies, we saw that the donor stool, if we put it in mouse, in the animal models, and we create avatar mice, they can actually induce a response to immunotherapy, which further mechanistically validates the importance of the donor microbiome in sensitizing tumors that they would be refractory to immunotherapy to these drugs.